Share the screen. Should be ready to go. Test. All right. So good morning, everyone. I hope you already went through the last lecture. I agree, it's really a heavy one. So we've been through, I think, around 6 new algorithms for indexing, which is something challenging. It's not an easy task. But I hope you will digest this material with the next programming assignment. Without doing it practically, it will be hard to understand how it works. And for today, we'll continue with a different topic. This is an extension of indexing. So we index the reference genome, but now what is the next step? What should we do with those indexed seed? And we agree in the first 2 lectures that sequencing is really a problem that causes those pieces to be individually separated from each other. And we need to put them back, which is the thing that we try to solve, which is a puzzle. And to solve those puzzle and try to connect all those pieces together, we need a reference picture. We need to look at the reference picture and try to decide what is the location of each of these single pieces. But until now, we didn't discuss how we do that. We agreed that we need to solve the puzzle, but how we do it. In the first lecture in indexing, we discussed that, giving this example, you have a reference text over here, and you'd like to find this query inside this text, which is exactly same as you have single piece, and you'd like to find it in a reference picture. And we said we could go sequentially scanning the entire text, and we compare character by character. If it is matching, then we move to the next character. Is matching the next until we find the entire sequence over there. And that is not an easy task because we may find really several occurrences in the same reference. We could do this easily in a notepad text editor called brackets. So I use it myself. Imagine you have the reference sequence over here. You have the FASTQ file over here in the same file. And then you double click on the read sequence, and then the editor will try to find all the matches in the entire file. So here, I just double click on this entire sequence, and I find many occurrences in this. And you already have those 2 files. You have the FASTA, which is the dummy FASTA file. We already attach it in the syllabus, so feel free to try it yourself. And we have the FASTQ. But the problem that you need to put both of them in the same file. This is how it works. Now, if you make any single change somewhere, you cannot find it. So it only can find the exact match, the entire thing. If there is a mismatch over here, you could click somewhere before that mismatch, and then you will find only that subsequence in the entire file. And we agree if we want to do that in a hard way, which is a brute force. Brute force means you are trying every single solution. Every single location. You're trying, does this match with this location or not? That is a brute force. So think about, you have a query, you scan sequentially, as we said, and that using an exact match. Exact match means we require the entire sequence to be exactly over there. And we said that is an expensive approach because it requires O, which is big O notation, for M and K. And what is M? You can match the color and guess it yourself. It's the query length, and then N is the reference length, and K is the how many queries we have. If we want to do that 1 time, 2 times, 3 times. But we could do that inexactly. Inexactly mean we use those expensive algorithms that try to figure out how many mismatch, how many insertions, how many deletions. And that will be really expensive, much more expensive than the previous approach, because now it's requiring quadratic time to find the inexact matches. And we'll go over some of those algorithms that do the quadratic time analysis, which we call them edit distance algorithm. All right. Now, solving that puzzle is done normally using a tool called ReadMapper. So a read mapper is the thing that we're trying to develop during this course to try to solve that puzzle, put all the pieces together in order to get back our reference genome. Okay. Now, what is read mapping? So read mapping is a computational step. Let's agree on that. Computational means it's just algorithm, and then we develop as software or hardware implementation, like using FPGA or GPUs. So it's not related to the wet lab. We're not doing any chemical reaction over here. Okay, so it's computational step that locates. So we're trying to find the location of every single piece, locates possible subsequences of the reference genome sequence that are similar to a read sequence. And the read sequence is the thing that we got from the sequencing machine. And that is our query. So we try to query the reference genome and try to find where's the possible location of that piece. Okay? While allowing at most E edits. And E is a threshold, which is integer number. And we call it edit distance threshold. Why we need to allow for some mismatches? So finding exact match will not help us. We really need to allow for some diversity. Again, the reference genome that we are using is coming from 2003, which belongs, 70% of it belongs to some random guy who resides in New York, right? And we use it for the entire world. Imagine that we are comparing African people, European, Chinese, American, and so on. All those diversities, we are comparing just for a single reference genome. That is bad, right? That's why we really need to allow for some mismatches, because by nature we are different. Even you and your brother, although there is 99.999% of similarity, but there's still some differences, right? That is one. The second thing, there are genetic diseases. And we talk about this in the first two lectures. Who remembers OpenSNP or OpenSNP database that we talk about? We already discussed it. We discussed the personalized disease, where you check this database, you check particular SNP or variation in your genome, and we check what kind of disease this might be causing, and what kind of personalized medicine would cure that disease, right? So OpenSNP is related only to single variation, single nucleotide polymorphism. So imagine you have your genome, you have your brother genome, you have some disease, or you may just have phenotype. Phenotype doesn't necessarily to be a disease. For example, eye color, hair color. You could have brown hair color, but your brother having black hair color, right? So it might be caused by genetic variation. If it is a single variation, we call it SNP, single nucleotide polymorphism, okay? So there is a database called OpenSNP as one word. You can try it yourself, it's for free, it's publicly available. You can see a list of all those single variation and what kind of medication we could use to cure this disease, okay? Now, because of that, we really need to allow for some differences. The problem that these differences not necessarily just be substitution, just replacing one character by another, but could be, so we have the substitution, we have the insertions, we have the deletion. And sometimes we refer to those as indel, insertion deletions, okay? So you could have some inserted character, just addition. So think about, you are breaking the sequence, you pull it aside, and you add some character. And we took some example where we add 500,000 character to one genome, which cause autism, right? Or overweight. So those two diseases are caused by large insertion, 500,000. Imagine how large it is. 500,000 added character, okay? We already talked about this in the first two lectures. All right, now tolerating a number of differences is essential for correctly finding possible location of each read due to sequencing errors and genetic variations, right? Now, but Now we agree on the definition of read mapping, but how does it work? First, we need the reference genome, which we call it FASTA format, and we talk about how this is structured. You already have the first programming assignment where we try to develop something to read this quickly, as long as you have also the query file, which is in FASTQ format. And we also talk about the structure of this file. Now, this file, we consider it as a list of queries. So those are the things that we should find in this reference, okay? How we do that? First, we need to build an index, a huge index that we could use to query the reference genome. Why we do that? Or the question another way, what if we don't do that? Okay? What if we don't do the index, which is the hash table? Do you remember the hash table? We've been talking about this for about 3 weeks, which is 2-column thing. We have the seed in one column, and then the location in the other column, right? 5, 7, 10, 400, and here maybe 500, and so on, right? That is a hash table. That is one way to build the index. And there are many other ways to do that. We don't necessarily need to use the hash table. Now, what is the benefits? What is the benefit of using hash table? Yeah. Go ahead. Think about if you don't want to store this location. We have a query, right? We will go sequentially, searching character by character, finish until the end of the reference. Now we got another query. What we will do? We will go again, searching from the very first character, try to match every single character until the end. We got the third query. Now we want to go to the beginning of the file, try to match character by character, sequentially scanning every single character. This is really huge, right? Instead of doing that, which is a very inefficient way, we just store some signatures, some pieces of this data, and we have it here. Whenever we got a query, hopefully some of those seeds that we already store here will appear in the query. And then we got that seed, we just search in the hash table. And one nice property of the hash table is that you can search it always with O1. That is the complexity to search the hash table. Why? Because this is normally sorted. So you already know where exactly to go to find it, right? You could do binary search, for example. You go to the middle and say something like, if the number I have as a query is greater than this, then I should search somewhere here. If it is greater than this, then I should go with this, which is always half the space. But if it is less than that, then I would go to half the way over here, and so on. Because I know it's sorted, so it's saving me a lot of effort, a lot of time, right? So that is very nice property of the hash table, or indexing in general. That's why we really need to use an index data structure, okay? Now we collect those pieces, and now hopefully we agree on the benefits of doing that. We do the exact same thing to the read sequence. And now we would like to query each of those pieces. And we discussed last time that it makes more sense if we have those seeds in the same order. We would like to have them as well in the reference in the exact same order, right? Because it makes more sense to have the blue followed by green followed by red. If those are swapped, then probably this is different sequence. Then we cannot claim that this exists in the reference. So we do the exact same thing to the reference genome, and we decide that this is probably the best location that we could find in the reference that's related to the read sequence. Now, how we do that practically? So we talk about indexing, where we have the reference genome, we extract some seeds or k-mers, right? And we pick some of them. We now have really large number of methods to do that. We build the hash table, and now we go to the next step, which is the lecture for today, which we call seeding. So indexing, we said it's related to the reference genome. It's related to building the index. That's why we call it indexing. So we need to store something in the hash table. Seeding is doing exactly the same thing as we do with the reference genome. However, there are fundamental differences that we don't store these things. Once we extract them, we don't store them anywhere, okay? We don't store them in hash table. We might store them temporarily, depends on your program. If you decide to do so, that's up to you, but you don't have to do it. You can scan them in the hash table. As long as you generate any one of them, you directly go to the hash table and you try to figure out, does it exist? Okay. And we do that only for the read or the query. We don't do it to the reference genome. We do it to the queries. So seeding is related to the query, which is the read sequence. That is the FASTQ file. The indexing is related to the FASTA file, right? Now, once we are already sure that some of those seeds exist in the hash table, we try to fetch the location of that sequence. So we fetch the location from the hash table, and we got some potential candidates that are similar to the read. So maybe this sequence and this sequence are really good, similar to the read based on the location we got from here, because both of them are sharing a large number of matching seeds. And we will see an example of that, okay? Now, some other candidates, although there are some seeds matching between the read and the reference or the location of the reference, but there are large other differences. Although we got, like, for example, we have a subsequence from the reference genome. We have the read over here. We got some matching seeds here and here, but how about the rest? For example, we have 10 characters, 10 characters, 10 here, 10 here, but this of length 200, for example, and this is 200. So we have only 20 characters out of the 200 matching between the two sequences. But we have about 180 other characters that do not match. Can we call this a good match? Can we call this high similarity between the sequences? No. That's why we require a third step called pre-alignment filtering, which try to quickly have a look at the two sequences that we identify by querying the hash table. And we say, oh, this doesn't look like good match because the ratio of matches with regard to the total length is really high. Or vice versa, if we look at the dissimilarity, the mismatches, okay? If so, we just ignore those candidates, and only those sharing large number of matches will pass the filter, and we go to the step called sequence alignment. Sequence alignment. We will have a bunch of lectures on sequence alignment. It's a very important topic. Those algorithms try to identify three important things. Yes, please. Step C. No, those are not seed. Those are the sequence. Those are longer than the seed. Longer than the seed. So we got the query, which is the read. We got the reference genome. We extract some seeds out of them, and then we try to match them using the hash table. Let's say we got 1 and 3 and maybe 2 matching between the read and some location in the reference genome, probably here because we have 1, 3, and 2, right? So we have 3 seeds matching between the read and the reference, right? Now we extract that portion from the reference and we try to have a thorough analysis with that particular subsequence. Why? Because we only have shared k-mers or seeds with that portion. Why we should look at the entire reference genome? We don't do that. We just pick the first number of characters from the reference, and we try to have it here, and we get the read over here, try to compare them together, okay? Which is this scenario. That is the read. That is subsequence from the reference genome, not the entire reference genome, subsequence, okay? And now we look at the number of shared k-mers. If it is 2 and the total length is way less than the total length of the read, that is something bad. If the total length of these seeds together, again, these seeds could be overlapping. It depends on the method that we choose. So if this seed is this way, so we cannot have the total length combined. We have to say this area is covered by maybe 12 characters. Although this is 10 and this is 10, but they are overlapping. So only we have 2 characters here extra, so that is 12. So if it's really 12 matching character out of the 200, still bad. Okay? So we'll look into that in detail. Hopefully this is clear. Now let's talk about the 4th step, which is sequence alignment. So sequence alignment is doing really 3 important things. Now, using indexing and seeding, I could identify a potential subsequence in the reference genome, okay? But until now, I don't know how similar and how different they are, right? I just know that they share some similarity and they share some differences. But how much, I don't know. That's why I need the 4th step, which will tell me how many. What are they? And what else? Where are they? How many differences do I have? How many differences between the 2 sequences? What are they? What type of differences I have? Do I have substitution? Do I have insertion? Do I have deletion, right? I need to know the exact type because each type might cause different disease. So I'm really interested in knowing that type. Where are they? Are they at chromosome 2 in location 10,000, 20,000, 200,000? The location really matters, right? Because it might impact something else. It depends on what gene that is impacting, right? So those 3 questions will be answered by that computational step, okay? Which is sequence alignment. That's why we are going to spend some time in that step. All right. So now let's see this in practice. I like always the examples, which can clarify many things, many questions you might have in the head. Now I have a read sequence. This is coming from sequencing machine. Let's agree on that, regardless if it's coming from Illumina, PacBio, or ONT, okay? Now we have a reference genome, and I pick in particular this example because I know that this sequence already exists in here and also exists somewhere else. So it exists in 2 locations, the reference genome. But now let's see how a read mapper could identify this location, okay? Now the first step is indexing. Now by this time, it's already boring. We already went through hundreds of examples, many algorithms over the last 3 lectures. So, but let's agree that we need to build kind of indexing structure. We call it a hash table. This is the seed, and those are the location. Done. And this is one example. In case you messed it up, what we need from this is the hash table. So if you missed this lecture, go back to the 3 previous lectures and try to watch those algorithm, how it works. But remember, this is what we need, the hash table, okay? Now let's move on. Those are the options that you have currently, and you will be using them in lab assignment 2, which will be released next week. So you have really large number of those. The first one was in lecture 6, the next 3 in lecture 7, and the next, which was the heavy lectures last time, lecture 8, okay? And it's up to you to choose any algorithm out of this list to apply it in your lab assignment. Now let's move on. Step 2, called seeding. Seeding, okay? Let's differentiate between indexing and seeding. In seeding, the first thing, we should bring in the read sequence, not the reference genome. The second thing, we should apply the exact same algorithm that we use in indexing. Whatever you use in indexing, you should use it exactly as it is. Don't choose different algorithm. It won't work, okay? If you use overlapping seeding, you should use overlapping seeding and seeding, okay? If you use minimizer, you should use minimizer and seeding, okay? Now here in this example, I choose minimizer, okay? And for minimizer, we need two parameters, K and W. K is the length of the seed or the k-mer, okay? W is our search space, where we should start working. So if there is 100 of those k-mers, we should pick only 5 of them and start looking at them, okay? We said in minimizer algorithm, we start to sort them in lexicographical order, meaning that alphabetics, but we start with the first character. If it's the same thing, we move to the second and so on. Now, this is easy example because only this one starts with A. And A precedes every alphabet, right? So this one should be in top of our list. That one will be taken as a seed out of this list. So we bring that seed, we have it somewhere, but we don't store it in a hash table because this is seeding step. We don't store it, okay? What do we do with it? We use it as a query, and we ask ourselves, does it exist in the index? Does it exist in the hash table that we just built? As a reminder, this is the hash table. Does this exist here? Yes. Where? At location 0 and at location 6. Okay? What does that mean? It means this read sharing similarity with the reference genome at those two locations. Okay? Now, what we do next? We're still in seeding step. We didn't finish seeding yet. Okay, it exists. That's great. Now, there is one important note. This is a read sequence. This is a reference genome. In the previous example, it was easy one. We got only 2 matching seeds. But in real life, unfortunately, we don't get that. What we get is this. Let's see. So we have here a read sequence. We have 3 seeds. So those colors are seeds. And this is means don't care. Could be anything, literally anything, okay? But only those 3 seeds that extract from the read sequence. Now, those are the matching seeds between the read sequence and the reference genome. So this seed, the blue one, is matching at this location, that, and so on. Likewise for the red and the green, okay? Now, this is a much harder problem than the previous example. The previous example is just single seed is shared in 2 locations. But here we have 3 seeds shared in really too many locations. What do we do with it? Which one can be a good location? We already saw an example. You remember the slide where we have a FASTA file and FASTQ file in the same slide? Try to match based on the color order, right? Let's do the same thing here. Can you guess what is the best location here? First one. Okay, what else? Any really nice candidate? Middle one. Here. Last one. Here. You have really matching. So we are missing the red, right? We have red, blue, green. So probably, this one is a good candidate. This one could be good if we are missing this as a deletion. So if it's deleted, we could consider that, right? What else? We have a blue and red. We have blue and red? No, we have blue and green. So now the red is deleted, right? So we consider that if we are studying a disease that is caused by large deletion, then we could consider that, okay? So now, this is the answer. Ideally, this looks really similar to what we have in the sequence. We have the blue followed by red by the green, but over here, we have totally different sequences. Now, this is up to some extent. It's up to us to identify that threshold. Do you want too much differences? Do you want little differences? Yes. Excellent. Yeah, that's a really great question. For example, this case, can we consider it a good one? How about the other one over here? Can we consider it? So we already have the red, We have the blue, we have the green, but in different order. Can we consider that? Now, that depends on the disease we are studying. This one could be really valid. This one could be really valid. Why? Although the red is swapped with the blue. Now, there is one type of structural variation. You remember structural variations? We talked about it, which is a variation that is longer than 50 characters. For example, 100 characters are deleted. This we call it structural variation, okay? Or 60 variations, 60 changes. Anything above 50, we call structural variation. If it is less, we call it just edit, okay? If it is single, we call it snap, right? Now, one type of those structural variation is to swap, to do swap in the genome itself. So there are some disease caused by swapping some portion of the genome, okay? In this type, if we are studying this disease, then we could allow for this guy, but not the others. Back to your question, now the distance between those seeds is also an important factor. Although they are in the same order, so we don't have issue, but how about the distance? How far we could go? That is something you really face in your lab assignment, okay? Excellent. We could go for 0. If we are looking for small variation, it doesn't make sense that here we have 100 distance and here we have 50, for example, okay? Now, it depends on two things. How many seeds you extract from the read. If you extract one seed and the length is 200, you extract 10 characters, you cannot allow for small distance because you still have large area you didn't cover by any seed. So you should allow for large distance, right? If you extract 100 seed from the read, then you have really dense number of seeds, right? You are covering almost the entire seed, the entire read, sorry. So you cannot have large distance between those seeds. That's why you have that threshold very minimal in this case. So it depends on what algorithm you pick. If you pick overlapping seeds, you expect to have too many seeds, right? In this case, I wouldn't allow for large distance between the seeds. But if you pick, for example, which one is providing the minimal number of seeds out of the list? Let's go back quickly. Out of this list, who remember which one provide the minimal number, which we call it coupled seed? Coupled mean linked together. So each two, we link them together. Which one? Stropmer's. Excellent. So Stropmer's was providing the minimal number of seeds. So in that regard, we expect to have very few number of seeds extracted from here. Then we would allow for large distance between the seeds. Okay? Maybe we should have +1. Write your name in the paper, please. Right? So we said that this is probably the valid location, and those are invalid based on some of our assumptions. Okay? So make sure you make valid assumptions in your lab assignments. Okay? Now, we identify already this location as valid. So what we do? We pick a subsequence from the reference genome over here, for example. We don't pick starting from this seed or this seed. Why? Because we are not sure if we collect seeds from the beginning of the read or not. We might be missing some characters over here. That's why we take some gap, or we allow for some large variation around the seed. So if those seeds covering 100 characters, we allow for 120, for example, 10 from here and 10 from there, and we take all this large subsequence. Some other tool, like Minimap tool, practically, they impose a restriction, saying that this blue should always start from the very first character, okay? So if this seed match with this read, then I'm going to always pick from this character. I don't allow anything over here, all the way forward with the read length plus some margin. So if the read length is 100, then I'm going with the very first character over here and pick the next 100 character plus some margin, okay? I allow for some variation to happen at the end over here. So that is Minimap 2 approach. But in other methods, other tools, they allow also for variation at the beginning over here. So they could take somewhere here 10 characters, and then they move forward with 100, and then another 10 characters over here. That's up to you in the lab assignment, okay? But hopefully we understand why we do that, why we allow for some variation over here and over there, okay? Now, how we would know that this could be a good location in practice? In this example, yeah, it's very nice. We could look at the color and then we could correlate them, right? But practically, we have only text file, which is FASTA. So we don't have those colors. How we would do it? So basically, when you have a query, when you have a single query, this is coming from the read, right? Read seed, let's say. And that's the hash table coming from the reference genome. We queried the seed in here, and we got the list of locations. So we got all of those, for example. Now we got another query. We queried the hash table, and we got those locations. What we would do, or how you will do it in lab assignment, you will bring this list, you'll bring this list all together and sort all of them together. Okay? Although they belong to different seeds. But we would bring this list all together, mix it up, and try to sort it. Why would we do that? Look at this example. If we now sort all these seeds, we would know that this seed is close to this. And it's followed by this, right? Followed by the green. Now, giving that information, I already know that those three are next to each other. And now I'm having a threshold called distance. I could know how far those three from each other, right? By knowing that, and if they met my criteria or whatever assumptions I have, then I could choose that as potential location. Okay? Now, because I'm sorting the list, I could know that this is really far from this. Then I would start again my for loop. I would start from the beginning of my algorithm because now this distance is exceeding. So that was one candidate. Let's ignore, move here, and start over. How many nearby seeds to this blue? Now I see the red is close by, assuming the threshold is already satisfying the distance over here. I need to check what is the third one. Oh, the third one is very far. Then let's cancel this. Now I go to the next in the list. Oh, it's a blue. All right, that's a great, matching the first one. Now, what is the next one close to that? That is all happening because I know the location and sorted. So sorting is helping me a lot. Now, arriving to this, the next closest one over here, it's really far. So this is invalid mapping, right? And this is how I would do it practically. Okay? Any question? Whatever, 100 valid locations. I will pick all of them. All of them. No, don't stop. That's why we have the third step, which is filtering. I'm going to filter those locations. And even after the filtering, I have the sequence alignment. It's going to give me the answer of these three questions. And based on this, I would decide which one is the best. Normally, I need one best, okay? But until I arrive to that conclusion, I really need to do some rigorous calculation. I cannot just intuitively say that this is the best, okay? I have to do some rigorous analysis. So hopefully this is clear how we'll do it in practical. Because this is due will be next week. Yes? Yes, excellent question. Now this belongs to this seed. This belongs to that seed, and this seed is real different from that seed. How I would know that this is blue and this is red? That's an excellent question, okay? So ideally, I would store them somewhere in 2D list, okay? So I would say here, this is the seed, this is the value, seed, the next location, maybe 2, seed, next location is 3, next location is 100, for example. This is how it looks like. once I sort it. So I sort based on this column, but I maintain the seed content. Why I need the seed content? To know if I'm moving forward or I'm still in blue. What if I got blue and then blue? I should ignore, right? I should move to really another seed. Okay, that's an excellent question. Do we have questions? Okay. Now going back to our easy example where we identify two locations. Do you remember what was the location? It was 0 and 6, right? 0 and 6. Those the location of the seed matches. Now this is 0 and this is 6. And this is the read. And what we extract is only a seed out of it, right? So we extract this seed, we try to match it. This seed ends over here until here. So we have 3 characters more. Now we match this seed between the read and the reference, and we got those 2 locations. Now what we do with it, we extract the same amount of character from the read and the reference, and we recognize it as 2 locations. So we have here, we have there. But remember what I told you, we should allow for some margin. So if the read sequence length is 10, we shouldn't pick 10 characters from the reference. We should allow for some margin, let's say 15. So this would be from here to here. And this would be from here to here. Okay? Why we allow for some margin? Because we could be studying insertion or deletions, right? Insertion, if I insert something in the middle, I'm going to push the boundaries. So this is going to be extended because it will be this way, right? And that's the inserted piece. I'm going to push the boundaries of the read and make it longer. That's why I'm allowing for some extra character to be included, right? Otherwise, I won't see this picture, right? So normally, they represent it using this way. This is the read. Oh, no. Let's have the exact same example. This is the read. This is the reference genome, what I have. Normally, they say something like this. You'll see this picture everywhere in Google Images. If you search for images and try to search for insertion or genomic insertion. Normally, they represent it this way, meaning that half of it is shared with this location, the other half is shared with another location, and there's a gap between them. That is the insertion. And this gap could be, if we are studying autism, for example, I told you, what is, well, I already erased it, it's somewhere here. You see 500,000. So there is 500,000 inserted character. If I'm studying this kind of disease, I should allow that in my read mapper. I should allow for this a huge gap, okay? If I'm studying just the snaps or single variation or small variation less than 50, then my threshold should be small. So there are traditional methods. If a known disease well studied before, there are a lot of research papers that could give you this insight, right? So you don't need to reinvent the wheel. You already know that this threshold in advance. But if you are studying something, for example, cancer is really different from one to another, and those mutations will be different. Some cancer are really complex. You cannot understand what kind of variation is causing that. So that you should take in consideration too many combination of those variations. You should allow for small, for large, for maybe something here and something somewhere else in another chromosome. That's happening, okay? Right? So now we consider this sequence. In the lab assignment, probably I will tell you this insight. I think I shouldn't let it up to you because it will be really difficult. So each student might consider 500,000 as threshold. Some other will consider it as one variation, right? Maybe I should specify that in the lab assignment, how much far you could go, okay? There's no max. It's up to the disease. Yeah, but if you study autism and you allow for this much, what about the other disease?
But if you don't have this 500K, then okay. But if you don't know what it is, then
No, it's not okay, because you are comparing to the entire genome, right? You're not comparing to the gene that causes autism. You're allowing everything to be 500,000. That's the problem. So you're allowing, and this gene, the distance could be 500,000, meaning that you allow for the blue and red, and the green could be here. So you could consider this as part of this, and you consider this as valid mapping.
Making the code complex or making the performance like time consuming or like energy.
You have to be careful about that. Now you already sort this list. Now, if you allow for 500,000, you should not check only the nearby seeds. You should check 1 and 2, and you could jump somewhere at the end of the list and you check. If there is no match, you will go back to here. So going back and forth is always expensive. So you have to be careful about it.
Yes. But it's like changing numbers from the code, but it will take forever.
Oh yeah, if it is parameterized, you could change the value, definitely.
Yes.
Definitely, yeah. Excellent. Yeah, this is normally how we have it. In Minimap 2, you could specify that distance. So you could have it what we call parameterized or user-defined threshold value. Okay. Yeah, excellent. You could identify that value. But sometimes not a robust solution. You have really to do customized read mapper for certain disease. Excellent, that's one point. Plus, the sequence alignment, if you extend the search space, if you give me 1,000 long sequence, when I compare it with 100, it will be more expensive than if you compare 100 character with 100, because there's a quadratic time. So you have to be careful about that. If you give this the entire distance to sequence alignment, it will be really expensive. But if you have the insight that this portion is really exact match with here, this portion is exact match with there, you could do some heuristic. You don't really need to compare this with this entire thing. You could just compare this with somewhere here, and you already know that this is exact match with this and this. So you don't need to do any math over here and over here. So you could build some heuristics really dedicated to certain disease, okay? All right, now we got this location from the reference genome. We got another location from the reference genome. We have the same read sequence, but what we do, we would need to compare this read sequence with this and the same read sequence with that as well, okay? The pre-alignment filter will go one step further. We already know that those seeds exactly matching each other, right? How about those? So pre-alignment filter will ignore those matching and try to check those things. How many of them are matching each other, okay? That is what pre-alignment filtering will be doing. And we will take a couple of examples over the next weeks about pre-alignment filtering in specific. Now, the last step is sequence alignment, which is going to answer these three important questions. And we have example why we need edit distance. I'm comparing organization with operation. What kind of arrangement I could have between these two words. So I could have exact match between O and O, R and R, and Asian and Asian, right? But in here, I have deletion from here, and I have insertion over here, right? But I could also have different arrangement as in here. O and O, R and R, A, and Shen all together, right? And I will have deletion over here, insertion, and more insertion. Now, I could have really more than that. I could have three arrangements between organization and translation, which is this, this, and this. But the question, which one is the right one? Which one to pick? Should I consider this? Should I consider that? Remember, whatever decision I made is going to affect the interpretation. What is causing that disease? Is it insertion followed by deletion? Is it deletion? Is it substitution, right? It all depends on how I arrange those alignment. Now, that is edit distance algorithm. Everything I'm doing here is edit distance algorithm, which at the end will give me that the number of edits is 1, 2, 3, 4, 5, 6, 7. That's why the edit distance is 7. To solve this issue, I should use what we call sequence alignment. Sequence alignment. So edit distance is going to give me a number which we define in the top as the minimum number of edits, which is insertion, deletion, or substitution, needed to make the read exactly match the reference segment. So it means what kind of operation I should impose over here to make this looks exactly the same as that, right? That is the edit distance and should be minimum. If it's not minimum, then that's not edit distance, okay? In here, I have 1, 2, 3, 4, 5. So that is not minimum because this is 5, but this is 1, 2, 3, 4. So that should be the right answer. That one easy. But for here, 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. Both of them 7. Which one is minimum? Both of them. Which one to pick? I don't know, right? Again, to answer that question, we should use sequence alignment. The difference between sequence alignment and edit distance is that I specify the penalty for each type. Penalty mean it's a cost, integer number. For example, did I delete it? Yes, it's not here. Let me maybe erase over here. I said the 3 variation, deletion, insertion, and substitution, right? Now I said if it is deletion, let the cost be, for example, -2. Insertion is 5, substitution is 1. Based on those costs, I could do a metric and do some calculation, give me an equation, the cost times each operation, and then add all that number up. And whatever give me the maximum number, that will be the right answer. Meaning that this is match. Do I have match cost? Normally, you have it as 0, or it's up to you to set it up. Then 0 times 1 plus, this is deletion. So the deletion cost is -2. -2 times 1 plus -2 plus times 1 plus this times the cost plus this times the cost, and so on. The total of that number, I'll compare it with the total of this operation. Which one is minimum, which one is the maximum? It's going to tell me that this is better answer than this based on my cost, because this cost means preference. So I'm preferring, for example, insertion over deletion. I want more insertion rather than deletion. And that's what sequence alignment allows me to do so. And at distance, I cannot do that because all the operation cost is 1, regardless the type. All of those should be 1, okay? 1, 1, 1, 1. So I'm treating all of them equally. That's why I call it sometimes unit distance. Okay? Unit distance means the cost is 1 for all type of operation. We will go over this again when we talk about edit distance or sequence alignment in detail. We're going in depth in each of those algorithms, right? If you are studying a disease that's caused by insertion, you don't care about matches, right? You want to really there are a lot of studies on how to pick them. There are well-defined cost metric that you can use as is. Okay, some of them even correlate with the width of the insertion. Okay, you prefer to have insertion, but how far you could go with insertion? Is it 1 inserted character or 5 inserted character? That's we call it gap extension. Okay, so we could extend that gap. Same likewise with deletion. Do you prefer to have 1 insertion and then some matches, then 1 insertion and then some matches and so on? Or you want to combine all the mismatches together? For example, this. Here, I'm preferring to have that as single gap. But here, I'm preferring to have 2 gaps. Which one you prefer? Okay? You could use those costs to identify those preferences. Right? And normally, this is how we solve it, which we call dynamic programming table. That what caused the quadratic time complexity. quadratic times goes by 2 nested for loop. When we look into the algorithm, how it works, we will see that it's really going for loop, another for loop inside, and so on. Okay, that's the nested for loop. What caused this? Why we need nested for loops? Because we are going to iterate over the rows, right? So we need an index to go from 0 to here. And also within the same row, we go to iterate from the first character to the last character, right? So over here will place us from one row to another, over here from one column to another. We will see that in detail. After we finish the sequence alignment, we will get something called SAM format. SAM format. It's really important to understand those formats. So we already been studying FASTA, right? And then FASTQ, and now SAM format. Okay. Now SAM format, we have here a header, right? A header file from here to here. Okay. Now this is the beginning of the actual thing in the file after the header. Now this and all of those until here is one line. It's really one row, one line. Okay, all of this is one line, but because I'm shrinking the area, it's trying to overflow over multiple lines. But this is in reality really one line. Okay, and those are multiple columns. So this is column 1, column 2, column 3, 4, 5, 6, 7, 8, 9, all the way to here. Yeah, this is the end of 9, and this is 10. And you can predict somehow that this and this is something that we know in advance. What is it? What does this looks like? Excellent. That is the basic quality coming from FASTQ file. Okay. So this is exactly as we have it in the FASTQ file. Okay. And then we have the next column over here, the next, all the way until we finish that line. And here is another line repeating itself. First column, second, third, fourth, and so on. Now, what is the meaning of each of those columns? We'll go over it by time. Okay, we gradually start filling this SAM format. But if you are curious, I attached the manual here. That is column 1, column 2, 3, 4, 5, 6, and so on. It's really easy to understand. For example, this is a query name, means the read name. Do you remember the FASTQ? The first line, we have the query name, right? Starting with @ sign. That is the query name. We have a bunch of flags, and this is integer number. For example, if it is 8, you need to convert it into the binary equivalent of it. So something like 1, 2, 4, 8. So we have 1, 0, 0, 0, and here a bunch of zeros, for example. Okay, this is how we convert integer to binary. Right? Now, what does each of those bits mean? It's really hard to memorize it. So what I do normally, there is a website called Explain Flags. Okay, you go to this website, you just insert the number and integer over there and ask this website to explain it to you. It will tell you based on the binary format and which flag is 1, mean activated. Will have some of those marked. For example, this means the read in reverse complement strand. Okay, the read is in reverse complement. That's based on the flag. So even myself, I don't remember what those flag means. So I just go to that website, paste the integer value, you'll get the meaning of those flags. In here is the reference name. So do you remember the FASTA file? We have greater than sign and then the name. That will be copied over here. Because the FASTA file might have multiple chromosomes, so we don't know which chromosome we are mapping to, so that will be the chromosome name. Position means the mapping location in the reference genome based on those seed matches. Map quality, we'll go over it in sequence alignment. CIGAR likewise, those we'll study when We go over sequence alignment and many other flags that we might cover some of them. Okay? Any questions so far? Now, there is one important point that we should take care of, which we already covered twice in the lecture, which is called repetition or duplication. So we know that human genome has about 50% of it just repeated. Okay? It repeats itself. Means it is a bit hard when we deal with human genome. So if I know that this region is exactly the same as this region, then I would expect the same number of seeds to match here and there, right? Now, if we talk about 50% of the human genome is exactly the same, that means I'm going to get a lot of this location appearing everywhere, right? So that you have to take care of when you do the assignment, okay? Hopefully we won't deal with the human genome because it's always complex and real large. But that is something to worry about when you identify the best location that you might have. That's why we have this map quality will tell us that really this is the best location or not. We're going to see a range of numbers for the map quality. The highest, the better. It means this location with high confidence, I'm sure it's the most similar one to the read or to the query. The other location, although it's just duplication, so the same sequence might appear everywhere, but might have some dissimilarity between the read. That's why I'm not considering it. And I'm considering this particular location because it shared the most out. Yeah, so this is the paper, if you're interested, from 2001, which is claiming that we have about 50% of duplication in human genome. This number is really varies. For example, there's a paper showing that there is about 40% of similarity between banana genome and the human genome. Imagine a fruit share really similarity with a human. And as I told you last time, it's not really like chromosome 1 is same as chromosome 1 in banana. Otherwise, we would be fruits, right? How they identify that is that they just quantify the similarity between the entire genome and the entire genome. So they have genome of banana over here, and they have the genome of human. Now, if this region is same as this region, they quantify that. That's the problem when they say about 40% or 70% between this and this are similar. Sometimes it's really not related. So if I have same genome in chromosome 1 and in chromosome 20, doesn't mean anything, right? Because functionally, they are different. A variation over here will cause something totally different from a variation over there. So functionally, those two regions are different. But normally in this paper, they quantify that. Regardless the functionality of those region or genes, they just quantify them, and then they came to conclusion that 70% we are similar to this thing, okay? If you're interested in that, I could share some of those references with you. All right. Now we have other important challenges when you consider doing read mapping. The first thing, what is the edit distance threshold we should use, right? This is the question your classmate asked, right? What could be used over here? Should we allow for 10 differences, 5, 1, 2, right? Again, that depends on too many parameters. What score penalty should we use? There are already well-established score penalty over there. We could use some of them, right? And I can give you a bunch of examples for this. However, it's still a valid question. Is the one we are going to use valid for the disease we are studying or not? Which sequence alignment algorithm should we apply? Likewise with the indexing. So indexing, we already go over maybe 12 algorithms, but there are other maybe 40 algorithms, right? Which one of them should we pick? Likewise with pre-alignment filtering, with sequence alignment, all of those steps. So we have 4 steps, but each we have too many alternatives. And each paper claimed to be the best, right? Claimed to have the highest accuracy, the low execution time, low memory, the lowest storage, and so on. But when we do that in practice, we might be surprised that this is not what we expect. Which type of alignment should we perform? Now, the terms, forget about it. We are going to explain it soon in the upcoming lectures, which is related to sequence alignment. Do you remember that dynamic programming table? So we have 3 types of those tables: local, global, and glocal, or sometimes they call it semi-global. Okay. Now, one important thing to note, and we already discussed a little bit about it, which we call reverse complement. Reverse complement. Now, do you remember in the sequencing, anyone remembers reverse complement, the term? What does it mean? What do we do with it? Excellent. Yeah. So we just discussed it 2 days ago. You already forgot? Okay. You remember in sequencing, we said we have the double-strand DNA. One of them coming from the mother, one of them coming from the father, right? This is how our chromosome. How normally we denote it as that. This is the centromere, and those are what we call legs or tail. One of them really coming from the mother and the father, right? And each of those strands is double helix. Okay, each of those is double helix. For example, this is like this, really. And the other one is really like this. I'm bad at drawing, but you can imagine what we are talking about. Okay. Now, when we do sequencing, we split those from each other, and we even split those double helix structure into single strand. So we consider only one of this. For example, 1 or the black, and even this is 2-line, right? So this is super coiled around each other, right? That's how we form the double helix structure. We consider only one of them for sequencing, only one of them. So we have 2 alternatives from father, because this is complementing this. And we have 2 from mother, this is complementing this, right? And we send only one of them to the sequencing machine. So either one of those or one of this. And then we start getting the A, C, G, T. But the sequencing machine doesn't tell us this is coming from where. Is it from here? Is it from there? Although, yeah, most of the time it's really similar. If we want to get the other strand, just reverse complement, I could get it from here. So I don't need to know it's coming from where. But the problem, if I have read over here, and then trying to map it to here, it won't fit. It won't fit. Likewise, if I have a read over here, and I'm trying to map it over here, I might end up having the location here, not in here, because it shares similarity with this location, but not in here, although it's just reverse complement. How we solve that issue? Normally, to solve the issue, we need to know what name, what we call this issue. We call it strand bias. This is strand bias problem. It's a really valid one, okay? And we solve it by simply consider when we pick the seed, we should pick seeds from both strands, assuming we have both of them. Or although normally in the FASTA file, we only have one of them. But when we want to do read mapping, we should consider as if we have both of them together. So we have this, and we build from it the other one, and we index both of them, okay? So if we extract seeds from here, we should extract seeds from there and store them in a hash table. However, practically, we don't do that. Practically, we don't do that. What we do, this is a Minimap2 example. Okay, this is if you go inside the source code, you will find this piece or snippet of code. What it says, I'm going to pick one of those two k-mers, assuming this k-mer or seed coming from the same location as in here. I'm going to pick one of those two, okay, based on this equation. Which one is the minimum? Which one is the minimum? So that is k-mer 0 and this is k-mer 1, okay? This one from forward strand, the other one from the reverse complement. And based on this, I'm going to choose one of them. Z will be either 0 or 1. 0 if this is minimum. If this is satisfied, then 0, right? If this is not satisfied, it will be 1. And based on that, I'm going to choose one of them to be stored in the index. So sometimes, let's maybe add another one. So again, let's just take a copy of this. Ideally, I would do this, which is storing all those seeds and all those seeds. But in Minimap2, okay, I'm taking sometimes from here, sometimes from here, sometimes from here. Some other time here, maybe, sometimes here a little bit, right? So I keep swapping between the two and either store those or those or those or those, right? This is what I'm doing basically here, based on the value to be minimum. How we know which one is minimum? Again, minimum here is something subjective. So you could choose lexicographically. Which one is minimum in terms of lexicographically? Means if it starts with A, then that is minimum, right? Or you could convert into hash value. You remember the hash function? So you convert the seed into hash value, which is a number, integer. And based on those integer comparison, you could identify which one is minimum, okay? In the lab assignment, I'm not sure if I would ask you to do that. It's a bit complex. Although in implementation, it's simple, but the problem with interpreting those later on, how you're going to sort those and those. So I may skip that from the question. I may not ask you. The goal is just to envision how a read mapper would be without having a complete or perfect read mapper, okay? But do you remember in the last lecture on Monday, what we talked about in the last 5-10 minutes? Do you remember the two-fold minimizer, the two-fold faster, two-fold faster minimizer? Do you remember the paper? It was from 20 January, I think. The one we opened it all together. We opened the link and click on it. And it was published in January 2024. And there was a nice example where we have blue seeds, red seeds, and then there was an equation. We quantify how many A's, how many G's, how many C's, how many T's. We do the sum, deduct something out of it, and then we got a number. Based on that number, we decide which one to go. Should we focus on the forward strand or the reverse complement strand, right? That is another way to solve this issue, okay? Which is strand bias. So you already have that in the very last few slides in the previous lecture. If you missed it, please go over it, okay? So now you have two solutions to do that, but most probably I wouldn't ask you to do it in the lab assignment, right? So hopefully we cover the entire overall plan, but we will go back now and we go over pre-alignment filtering in depth and then sequence alignment in depth, okay? We will continue doing that over the next 4 weeks or so until we finish all those steps. Hopefully by covering the entire thing related to read mapping, you already envision what you are going to do over the next lab assignments. So the next step would be to implement an indexing step, okay? To pick one of those algorithms, it's up to you. I will leave that up to you. Try to implement it and pick some seeds from the FASTA file, store it somewhere, pick some seeds from the FASTQ file, don't do anything with it, don't store it. But later on, we'll use it to query the hash table and then identify some location in the reference genome. Once we do that, we go and pick one of the pre-alignment filters, implement it, apply it. One of the sequence alignment, implement it, apply it, done. Okay? Should be straightforward. I don't expect to have any issue. Sometimes you have some surprises. We will do some assumption. That's the best you could do, right? You assume something. I'm assuming this distance. I'm assuming this threshold. That's perfectly fine, right? There are other computational challenges, and this what makes your tool more popular than others, especially if you could handle long reads. Long reads is really challenging. So we have short reads and we have long. Long, when you try to extract overlapping seeds, you'll get thousands of them, 10,000 of them, million of them, right? How you deal with those large number of seeds? Same thing with memory. What kind of memory you'd like to require? Execution time and accuracy. We'll go over that. Part of them related to the indexing algorithm that you choose. Some other parts relate to the sequence alignment. Some other parts relate to pre-alignment filtering and so on. So each of those components contributes to the overall performance over here. And I show you that table, if you remember, based on the indexing algorithm. Indexing time will be different, and indexing main memory will be different, and even the size of the index will be different. That's the end of today's lecture. Hopefully, we cover some of the aspects that you really need relate to seeding and querying. Seeding, we didn't cover much because it's the exact same thing as with indexing. So the exact same algorithm you would use in indexing, you would use it with seeding, right? So that's why there's not much to say over here, but querying the hash table is the tricky part, which is something that you should really take care of. So we have the strand bias, we have also sorting the location, and we have also that distance to identify what of those regions might be a valid mapping or invalid mapping. Okay? Do we have any question? Can take up to 5 minutes or so. This one. Over I think this. Oh. Oh, is it the questions, those? Okay. It's one. That's why we call it unit distance, or sometimes Levenshtein distance. No, sometimes you are good with edit distance. Sometimes you are good with sequence alignment. Sometimes you are good with something really simple as Hamming distance. What is Hamming distance? Anyone of you working in network, computer network, security, privacy? No? Okay. Hamming distance is used for error correction. So, you know, when you send packets over a network, you might miss some of them. There might be some errors introduced in the packet. So how you would ensure that the entire packet arrives safely to the other destination? You would use something like Hamming distance, which is basically you do XOR operation between every single character. Okay? So if you have two sequences, you compare this and this, this and this, this and this, this and this. So ideally, you allow only for substitution. Or in the other way, you are able only to detect substitution. Okay? You cannot detect insertion or deletion. So you could allow for that to be used to replace sequence alignment. That's in case only you are studying SNPs. And there are tools that use only Hamming distance. You can imagine it's really fast. Just for loop, go over the character, done. So the complexity is ON, not ON quadratic, as in edit distance algorithm. So there are alternatives to sequence alignment. If you don't want the heavy one, the most expensive one, you could use edit distance, or there's something called sparse DP, sparse dynamic programming, which is something between, let's say, sequence alignment and Hamming distance. How it works, you have the two sequences, you have seeds over here. So you say, I know this is exact match with this is exact match with this, but I'm not sure only about this area. Then I'm going to apply edit distance only here. And the rest I can just tile together or stitch this area with whatever result I'm going to get here. Just stitch this with this and then stitch it with that, and then you'll get the sequence alignment result for the entire sequence. That's what we call sparse DP, because I'm applying edit distance only in certain region rather than to the entire sequence. Okay? There are a bunch of alternatives to sequence alignment. That's the short answer. All right? Okay. Thank you so much. We'll see you next week. Take care. Oh, thank you so much. Yeah, it's real nice to see the new faces today. So please keep it. Please try to coming every lecture. It will be really beneficial to you.